Hello friends, today we are going to study about composition, structure and function of proteins. In that we will study amino acids, especially the structures of all the 20 amino acids and their importance. Amino acids can be classified into three groups. First is amino acids with non-polar side chains. So under these, the following amino acids are classified. These are glycine, alanine, proline, valine, leucine, isoleucine, methionine, phenylalanine, and tryptophan. It is better if you remember the one-letter abbreviation for all the amino acids. For glycine, it is G, alanine A, proline P, valine V, leucine L, isoleucine I, methionine M, phenylalanine F. F from the pronunciation. If you pronounce phenylalanine, the sound is like F. So it is F, and tryptophan, it is W. So these are the list of amino acids with non-polar side chains. Non-polar side chains means they are hydrophobic in nature. These are hydrophobic amino acids. Now, what are the importance of hydrophobic amino acids? They are mainly found in the interior of the protein. Whenever a protein fold, because the proteins are present in an aqueous environment, the biological system is an aqueous environment. Therefore, the proteins are found in an aqueous environment. Proteins which are soluble in the aqueous environment, in these proteins, all these non-polar amino acids or the hydrophobic amino acids are present in the interior of the protein. So therefore, they play a major role in protein stabilization by hydrophobic interaction. Because of these amino acids only, the protein can fold in a particular way and can perform their function. So the importance of hydrophobic amino acid or a nonpolar amino acid or the amino acid with nonpolar side chains are, these help in protein folding and help to stabilize the protein in a folded conformation through hydrophobic interactions. For a soluble protein, if we talk about a transmembrane protein, like transmembrane protein means this is a membrane. If any protein is present like this, these are known as transmembrane protein. So this part is soluble, this part is soluble, but the part which is present between the lipid bilayer should be non-polar in nature. So this part mainly consists of non-polar amino acid or the hydrophobic amino acids. Mainly, I'm not saying that all the amino acids present between lipid membrane all will be non-polar. Maximum amino acid should be non-polar. Some will be polar but uncharged. So more is of non-polar nature, some is of polar but uncharged. What all we can see with these structures? If you see the structure of glycine, it is H, simple. If you see the structure of alanine, CH3, it's simple. So glycine and alanine, these are mainly found in the beta sheets. In the upcoming videos, we'll study about the secondary structure of the protein. The secondary structure of the protein consists of alpha helix and beta sheets. In beta sheets, it is generally 6 to 22 sheets are present like this, and they're bonded with each other. Because there are less space in between, so mainly it is composed of glycine and alanine because they have a short R group. Next is, because glycine is having H as an R group, so it is very flexible in nature. Because of its flexibility, these are found in the turns and twists. Wherever glycines are present, from there the protein can turn. It can twist very easily. So mainly the glycines are found in the turns. Coming to proline, because of its ring structure, it gives rigidity, steric hindrance, right? So these are also found in the turns to give them rigidity. Coming to phenylalanine, contain a benzene ring. Tryptophan, it also contains a benzene ring. So these two are the aromatic amino acids. If you see the structure of methionine, it contains sulfur group. But the sulfur present in case of methionine is not available to bond formation. Now, phenylalanine and tryptophan, these are bulky amino acids because they contain heavy R groups. In a protein, the bulky amino acids cannot be present together in a long stretch because they can create steric hindrance. So never in any protein, the bulky amino acids you will find in a long stretch. So these are the amino acids with nonpolar side chains with their importance. One more is, there are two types of protein. One is fibrous protein and one is globular protein. Fibrous protein, the example is alpha keratin. Where do you find keratin? in your nails, in your hairs, right? So these are water insoluble. That means these are hydrophobic. So that means these proteins are composed mainly of these non-polar amino acids. So fibrous proteins are mainly formed of non-polar or hydrophobic amino acids. So depending upon the property of the amino acid and its structures, we can easily find out the property, structure and function of a protein. Next is amino acid with polar but uncharged side chains. These amino acids are serine, threonine, cysteine, asparagine, glutamine and tyrosine. S, T, C and Q and Y are the single letter abbreviations for these amino acids. Now, if you see the structure of cysteine, here also a sulfur group is present. In the last slide, we saw in methionine also sulfur is present, but that methionine is not available for bond formation. But in case of cysteine, this sulfur is available for bond formation. Therefore, cysteines are involved in disulfide bond formation. That means one cysteine here can form bond with another cysteine. But to form this bond, the H present here should be eliminated. Removal of hydrogen is oxidation. So cysteine should be oxidized to form disulfide bond. Therefore, the proteins that are secreted outside are composed mainly with cysteine. Why? Because the interior of cell is reducing environment. The exterior of the cell is oxidizing environment. So the proteins that are secreted outside the cell contain more of cysteine because the cysteine can involve in disulfide bond formation and can help in stabilization of those proteins. The interior environment of the cell is a reducing environment. The exterior, the extracellular environment is an oxidizing environment. In the oxidizing environment only, the cysteine can form disulfide bond. Therefore, the proteins that are secreted outside the cell contains more of cysteine so that they can form the disulfide bond and can help in stabilization of secreted proteins. Tyrosine, one more aromatic amino acid. In the last slide, we saw it is phenylalanine and tryptophan. Here, tyrosine. So these three in total are the aromatic amino acid. Understood? So these are polar uncharged. Correct. Non-polar amino acids are found in the interior of the protein. Polar uncharged amino acid are found in both exterior and interior. Polar charged amino acids are found on the exterior surface of the protein. Non-polar amino acids are found in the interior surface of protein. Polar and charged amino acid are found on the exterior surface of the protein. Polar and uncharged amino acids are found in both exterior and the interior surface of the protein. So these amino acids which are polar but not charged can be present in exterior as well as on the interior surface of the protein. Next is amino acid with polar charged side chains. So it includes lysine, arginine, histidine, aspartate and glutamate. These are the amino acids which are polar as well as charged. Why charged? Because in all the amino acid, this part is common. One positive and one negative. They cancel out each other. One extra negative. So these are negatively charged amino acid. Negative charge amino acid. Positive, positive, positive. So these three are positively charged amino acid. These two are negatively charged amino acid. Now, what is the importance of positively and negatively charged amino acid? Because these are positively and negative charged amino acid, they can play an important role in electrostatic interaction. In alpha helix, because it will be in like helical turn, so these positively and negatively charged amino acids should be placed in such a way that they can form ion pairs. They can form salt bridge or they can form electrostatic interaction and help in the stabilization of alpha helix. So the charged molecules help in the stabilization of the alpha helix because they can participate in electrostatic interaction. Apart from that, they found in the exterior surface of the protein because they are water soluble. They can form bond, hydrogen bond with water molecules. One more thing. If you see the structure of aspartate and glutamate. In the last slide, we studied asparagine. What is the difference in the structure? Here C double bond O, the structure here is C double bond O, O minus. In asparagine, it will be NH2. Similarly for glutamine also, C double bond O, here in place of O, here also NH2. So amino group is present. That means these amino acids can participate in transfer of nitrogen. Asparagine, aspartate, glutamine, glutamate, they can help in the transport of nitrogen. So these are all about the amino acids, the importance of the structure of amino acids and their properties to determine the structure, function, or the location of any protein. If we know whether the protein consists of more of polar amino acids or the non-polar amino acids or more of disulfide bond cysteine residues, so we can say the protein function, what could be the function of the protein, what could be the location of the protein, correct? So these how we can analyze the function and the structure of a protein or its location by studying the amino acid composition of that particular protein. Practically, we can use this also for protein engineering. What is protein engineering? That is the proteins which are already present, we engineer the protein to make it better. One example I'm giving you of protein engineering. You all know paper is made from wood pulp. Now, to make paper from wood pulp, chemical blanching is done. Because of this, the effluent from these industries are very harmful. So, if we can replace the chemical with any enzyme, it will become eco-friendly. So, the enzyme which is used in this process is xylanase. But the step in which the enzyme is used, it also requires high temperature. Now, in high temperature, the xylanase become unstable and they lose their activity. Now, how we can make the xylanase stable in high temperature? By adding cysteine residues. If we add cysteine residues, what will happen? More cysteine means more disulfide bond. And more disulfide bond means more stability. This how xylanase is made stable at high temperature. So this how xylanase is made stable so that it can tolerate the high temperature without losing its efficacy. So this is called protein engineering. We are engineering a protein to make it more better or more efficient in their function. So this how practically we use the properties of amino acid to make the function or the structure of protein much better, or we can increase the efficiency of their function. Now, one question from my end for you all. Ribonuclease from bull serum, which is a dimeric protein. What is dimeric protein? Having two subunit, you can say. So, ribonuclease from bull serum, which is a dimeric protein, it has an anti-tumor property and used for the treatment of tumor. But the repeated use of this particular ribonuclease produce antibodies in our body because these ribonuclease has taken from bull semen, which is foreign for our body. So our body start producing antibody against this particular ribonuclease. Correct? So what could be the better if we can use human ribonuclease, because those ribonuclease won't be foreign for us. But human ribonuclease is monomeric in nature. To function as an anti-tumor, it should be dimeric. So the bull serum is dimeric, but the human ribonuclease is monomeric. But the use of human ribonuclease is more beneficial for our body because our body will not produce antibody against the human ribonuclease. But it is monomeric in nature. Now you have to tell me how we can engineer this monomeric ribonuclease of human to make it dimeric so that it can function as an anti-tumor agent. So you have to tell me how we can make one subunit into two subunit. Correct? Which amino acid addition can do that? Do let me know in the comment section if you found the video informative. Please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe and stay happy.